All right, folks, let's try this again. Lance DuGoud here, doing my best to educate folks about things that maybe you don't know enough about. Might be some misinformation out there, and might be some things that you're just curious about. And I don't like to watch the news because usually it's just bad information, misinformation, and bad news in general. And I don't like being sad or depressed, but a lot of things out there maybe need some attention and need some redirection. So my very first of these was about type 2 diabetes. So if you know somebody with type 2 diabetes, you have type 2 diabetes, you might take a look at that, kind of get some education on that, and so maybe you know more about it and can live a healthier life or help somebody live a healthier life as well. This go around, it's going to be a multi-part series. This is part one of COVID-19 stuff. Just again, not a lot of people getting the right information out there, so I want to try to help everybody out, get everybody on the same page, and hopefully then bring us all to a healthier place in life. So viral basics here, you just kind of have to put up with my big old cursor, but did that on purpose so everybody can see it. Virus basics, things you don't know, might have been told or might have misconceptions on coronavirus. It is part of the common cold. There's respiratory syncytial virus. If you have any children, you might have seen or heard that. Rhinovirus and parainfluenza. So that and coronavirus all make up the big four of respiratory viruses that we see in human beings. It is a respiratory virus, not a gastrointestinal virus. So please stop buying up all the toilet paper. Seriously, there are families out there with children that are in need of toilet paper. Please share your surplus with your neighbors. Just doing the right thing, trying to help people out. In other species, in cats and dogs, coronavirus is in fact a gastrointestinal virus, but while quote-unquote experts might say that folks with COVID also had diarrhea, there's reasons why you would have diarrhea with a very severe systemic illness that does cause terrible fever and terrible inflammatory processes. So it itself is not a primary gastrointestinal virus, so just kind of be aware of that, and let's get off that bandwagon if you don't mind, because I do have a family as well, and I worry about us even having enough toilet paper. That's a silly thing to have to worry about. I think we can all agree on that. But back to coronavirus. It's been around for decades, if not centuries. This is a virus. Like all viruses are not alive. So no virus is alive, so it can't be killed. If your disinfectant says kills viruses, well, they're wrong. So don't listen to everything that you hear. Viruses can be denatured, destroyed, or deactivated. It can't be killed. Okay, so it doesn't live, floats around in microscopic dust or water vapor, little tiny particles in the air until it hits a very specific receptor. When I say specific, I think of a key in a lock. You can have a lot of keys, viruses, put into any old door lock, like your airways, but only specific keys will actually work on that door. So you have to have the right receptors for the virus to attach. That's why coronavirus is not an eye infective. It's not a skin infective. It's not a gastrointestinal virus. It is respiratory because its target is a respiratory system. So how viruses work, I'm not sure how I'll do this the most simple way, but I'll do my best. I got an image I want to show you as well. Again, receptors on the virus are binding to specific target cells. Okay, you don't get chickenpox in your eyeballs. Dogs don't get cat leukemia virus. Okay, viruses are very specific. Occasionally, they can cross species like the avian flu. Influenza viruses are a little bit longer discussion, a little bit different. But right now, we're not in a panic over the flu, which, by the way, is an epidemic in the United States annually. Okay, so when we think of things like endemic in a localized area like your town, epidemic across maybe the United States, and pandemic across the world. Well, let's face it, the flu is probably a pandemic virus as well, just with fewer life-threatening cases. I'm just making that point so maybe everybody stops using the word pandemic so aggressively, because let's face it, if you're not leaving your town, if you're not leaving your state, you really don't have to worry about the pandemics of it. You need to be worried about the endemics of it, endemics of it. So that being said, again, I just want everybody to have a little bit less stress in their lives and a little bit less drama. For me, I call it psychological gaslighting terminology. When everybody says pandemic this and pandemic that, let's face it, unless you're going to Italy or going to China, you're really not that concerned. You're concerned about maybe their health, but not that concerned about contracting something from them because you're not in that area. So anyway, back to the science. And so here we go. How viruses work. Sorry about that. And I want to show you the image here that I have. Very basic stuff. Human coronavirus is an enveloped virus with these little spike proteins, the green things on the outside. Those spike proteins have to sit in this little cup, the receptor that is very specific to that little green spike protein. If you don't have the receptor, it can't sit down. If it can't sit down, it can't infect you. Okay? So once it sits, then it sort of grasps this envelope itself into the cell itself, comes in, releases the RNA, and unlike some viruses that actually go into your DNA and replicate themselves, this one kind of stays out of the nucleus, the center of the cell, and replicates itself sort of in the cytoplasm, in the major section of protein production inside your cell. So you make from the RNA of the virus, you make the nucleocapsid, the membrane, the envelope, and the spike protein. So you're basically building these things. Okay, and then as you're replicating the rest of the RNA itself, then you're packaging that in one area, packaging all the proteins for the receptors in another area. Those things combine, and then it forms a virus particle. That virus particle then pinches itself off and then releases into your body. So one virus can come in and tens of hundreds, if not more, viruses can be formed and come back out. Now, this is a little bit simplistic. Occasionally along your membrane, you are the pink here, occasionally along your membrane, you will still have a spike protein or an envelope protein. You'll have things still stuck in there. So imagine if those little green things and blue things are still actually stuck in the cell membrane of your cell. That there is not good, and your cell doesn't like it, and a lot of times your body will attach an antibody to that, get stuck to that, and then burst your cell open, okay? Because it wants to bust it open and build antibodies to all this stuff. So your body is detecting the other little surface things that are stuck in here, okay? So it's not a perfect process, but in fact, you are getting some of those things still stuck in your cell membrane. When this virus releases, you're going to try to attack it with antibodies. You're going to try to attack it when you can, but when those surface proteins are on here, still stuck in your cell membrane, your antibodies are going to stick to that. And when it does, it's going to destroy this cell, and anything on the inside of this cell, including all this viral stuff, and your own healthy normal tissues, it's going to try to build antibodies against the foreign objects. When it does that and it ruptures this thing, you get a heck of a lot of inflammation. Inflammation causes fever, inflammation causes cough, and it makes things feel and act a heck of a lot worse. Okay, so I just want to give you the basics on that. Feel free to rewind it, review it. It's basic virology and a little bit of immunology for you too. So again, the virus itself opens a channel using those receptors, and the genetic material enters the cell. Coronavirus uses the RNA to form new viruses. Now, while it's making new RNA, it doesn't proofread. It doesn't make sure that the RNA it's creating is exactly the same as what came out. Give you an example here. Here's the stuff that came in. Maybe that's exactly the way the coronavirus intends to be, but when it replicates itself, sometimes it does it too quickly, and then this molecule is not the same as it is here, as it is there. And if it's not the same, that's mutation. Mutation is a good thing. A lot of people say, oh no, mutation is bad. No, it gets super bugs that way. Look, they mutate all the time. When DNA or RNA mutates, including in our own cells, when it mutates and changes, it's typically not correct, it's not proper, and it's not in a virus or bacteria. It's not going to be infective. If you mutate the DNA of a baby as it's a fetus growing inside of a woman, if you mutate that, a lot of times the child doesn't survive, or they come out with genetic mutations and flaws. So mutation, as far as infectious disease goes, is a good thing. So when the coronavirus doesn't do a lot of proofreading, it's mutation. And when it's released, that mutation sometimes makes it so that virus cannot then attack, attach, or affect the next cell down the line. Okay, so again, part G here, when the cell ruptures or releases that virus, or the body attacks that cell because it has those receptors stuck in its own membranes, you also release your own proteins, your own DNA, and your own RNA. Okay, that's really, really important in one of the later discussions when I discuss the testing for COVID-19, because a lot of these tests are looking for proteins, and they're also looking for our own proteins in DNA to confirm that our cells have been ruptured, to confirm that we have actually been infected. So we should see a rupture. We should see our own contents come out into those samples on the nasal swab and otherwise. So again, it's the cell rupture that causes our bodies to respond. The more cells destroyed, the more inflammation, the more cough, the worse the symptoms. Again, going back to mutations, if the mutation in viruses and bacteria occur, they're generally bad for them. They only have a certain amount of genetic material, and if they change something, they may mutate the piece of RNA that's actually encoding this nucleocapsid or a spike protein. If you change this spike protein, it cannot sit in the cup. If it can't sit in the cup, it can't infect. So let me just give you a basic example of this. If you have a spouse or a loved one or kids, have you ever had them get sick and you didn't get sick? Or maybe you got sick and they didn't get sick. And I'm not just talking about coronavirus. I'm talking about any old cold. You get the sniffles, you get the cough, you get diarrhea, you get the flu, you get something. But not everybody in the family gets it. Is it because you're just perfect at keeping everything clean? Well, you can't be because you got sick or the kids got sick. But the fact that you don't get it means that mutation happened, and that mutation caused enough changes that that virus came out of them not infective. Okay? So again, mutation is a pretty good thing. And then with little to no proofreading during that, the virus particles aren't perfect. Maybe RNA is not perfect, which leads to many inactivated viruses, which limits the spread of disease. So let's just say that 1 in 3 times that it replicates, there's a mutation that causes it to change enough that it can't reinfect somebody, that it can't come out contagious, that it can't even attach to a human cell. And that means if this thing lets out 3 of these, 1 of these is bupkis. 1 of these is bad. So that means though you're sick and you're spewing the viruses out, not everything that you spew out is contagious. So why have we seen a reduction over time in the number of sick patients and the number of deaths and things like that? It's because enough of us have had it, and whatever came out not infective. So what happens to those viruses? Well, they can't hit their targets, they can't infect, they can't replicate, so they just go away, and that's the end of them. Again, so you may have had a cold at some point, but your loved ones didn't get it. When it came out, it was deactivated. So again, in this case, mutation and change is good. All right. So let's see here. So key points to this little discussion is, remember, it's a respiratory virus, so please stop buying up all the toilet paper. It's a scary thing to think that you can't even wipe your own bottom, and I know that I'm not going to touch everybody's lives here, but it's a respiratory thing. It's not a gastrointestinal thing, so we don't need to be hoarding all the toilet paper. Please share with your friends and your neighbors. Mutation is good, not great in humans, but don't fear the change. Don't fear the word. Don't listen to the media when they say, oh, mutation, and it's going to be a super bug. It's going to be around forever. Again, the changes often inactivate many virus particles, making them less infectious, even non-infectious at all. Again, viruses are not alive, so we're not killing them. Another video, I'm going to tell you what we can do and what we are doing to help along the way, but we do have to be careful with the disinfectants and the chemicals that we're using. So viruses aren't alive, so don't read the disinfectant when it says, oh, it kills COVID-19. It doesn't kill COVID-19. Deactivates, denatures it, but it does not kill it. And I'm very particular about that because they know better. They know better than to use that terminology. They know what the word means to kill, but they use it because they think that you are not smart enough to know better. And now, after listening to this, you are smart enough to know better. So they better start changing that. Otherwise, I hope everybody stops buying the garbage that says it kills a virus. And then remember, again, and this is going to be important when discussing the testing available, cells release all sorts of things when viruses release, including our own DNA, RNA, and proteins, as well as the viruses that look normal but aren't functional. So again, not every virus that comes out of an infected cell is functional. It's going to be very, very important, mainly because when we are testing for these things, we're not testing for an actual functional virus. We're just testing for bits and pieces of that virus. So it all seems easy enough, so the next one then I'll move on to will be something new, how to stay safe and healthy, what we're doing, what we already have done, and how it's helped, or has it helped, because no one specific source for information seems to answer all your questions, but I'm going to do my best to hit all the major topics. Be well, be safe.